Okay, so today we are diving into something pretty mind-blowing. The potential of existing drugs for cancer treatment. You're interested in new cancer treatments, and we've got a stack of research articles here hinting at some surprising possibilities. Tell me, is this really a thing? Can we actually fight cancer with medications already sitting on pharmacy shelves?
It's a hot research area, drug repurposing, as it's called. Um, it's showing real promise. Essentially, we're finding out that drugs designed for one illness can have like a hidden talent for tackling cancer cells.
Like what? Give me an example. That'll blow my mind.
Okay, well, let's talk about nitazoxanide. It's typically used to treat parasitic infections, but this research we have here focuses on its impact on colon cancer.
Wait, hold up. A drug for parasites fighting cancer.
Yeah.
That sounds like something out of a sci-fi movie. How does that even work?
So, researchers discovered that nitazoxanide inhibits a specific signaling pathway called 1/catenin, which is often overactive in colon cancer cells. Think of it like this. This pathway is like an accelerator pedal stuck down, making the cancer cells grow uncontrollably. Nitazoxanide steps in and effectively releases that pedal.
So it's putting the brakes on this out-of-control growth. That's incredible. But it gets even better, right? There are other examples.
You bet. In fact, the study showed that nitazoxanide wasn't just effective, it actually outperformed the standard chemotherapy drug 5-fluorouracil or 5-FU in lab tests.
Wow, outperforming a standard chemo drug in the lab. That's a big deal. What's the secret sauce? Why is it better?
It comes down to how it targets those cancer cells. Nitazoxanide was shown to have a higher IC50 value against normal colon cells compared to 5-FU. Essentially, it was less toxic to healthy cells while still effectively killing the cancerous ones.
Okay, so it's more precise in going after the bad guys while leaving the good guys alone. That's really promising. Tell me more about these repurposed drugs. What else are we looking at?
Let's move on to another interesting candidate, ivermectin. Now, before we dive into the science, I know there's been some controversy surrounding this drug, especially during the pandemic.
Yeah, ivermectin definitely made headlines. But we're focusing on the science here, right?
Yeah.
What's the story with its potential anti-cancer properties?
Right, the science is key here. Studies suggest that ivermectin might be effective against several cancers, including breast cancer, by inhibiting a protein called PAK1.
PAC 1. Okay, for those of us who aren't walking medical dictionaries, what's so important about this PIK1?
Imagine PAK1 as a central hub, or a control center, within a cancer cell. It receives signals from various pathways and dictates crucial processes like cell growth, survival, even inflammation, all the things cancer cells hijack to spread and survive.
So by inhibiting PAK1, ivermectin is essentially cutting off communication to that control center, disrupting the cancer cell strategy.
Precisely. And what makes this particularly interesting is that PAK1 doesn't just impact one thing, it's a master regulator. So by targeting it, ivermectin could potentially disrupt multiple pathways that cancer cells rely on to grow and spread.
That's fascinating. So we've got this parasite medication potentially targeting a specific signaling pathway, and then ivermectin, a drug with a more complex history, potentially disrupting a whole command center within cancer cells. Are we seeing a pattern here in how these repurposed drugs work?
What we're seeing is that these existing drugs often target specific molecules or pathways that are crucial for cancer cell survival and proliferation. And the fact that they're already approved for other uses can significantly shorten the development timeline and reduce costs if their efficacy in treating cancer is confirmed.
That's the beauty of drug repurposing, isn't it? It's like finding a shortcut through a jungle because someone else already blazed a trail, even if it was for a different destination.
Exactly. And it highlights the interconnectedness of biological systems. What might seem like an unlikely candidate for cancer treatment might actually hold the key to unlocking new therapeutic avenues.
Now, I've heard whispers about other substances being touted as cancer cures, things like amygdalin, sometimes called vitamin B17. What's the deal with that? Is it legit?
Ah, yes, amygdalin. It's found naturally in apricot kernels and bitter almonds. And while some claim it's a miracle cure, the scientific evidence simply isn't there to support those claims.
So more hype than hope. That's disappointing, but honestly not surprising. With the amount of misinformation out there, is there anything to be concerned about with amygdalin?
Unfortunately, yes. Amygdalin breaks down into cyanide in the body, which, as I'm sure you know, is incredibly toxic.
Okay, that's a bit terrifying. It's a stark reminder that just because something is natural doesn't automatically make it safe. What about other natural remedies? Anything promising on that front?
Actually, there's some intriguing research on soursop leaves, specifically a fungus that grows on them.
A fungus. Now, that's not what I was expecting. Are you serious?
I am completely serious. And get this, This particular fungus extract has shown promising results against cervical cancer cells, specifically HeLa cells, in lab settings.
Okay, you officially have my attention. A fungus on a leaf fighting cancer. What's the story there?
Researchers in the study isolated this specific fungus that grows on soursop leaves and discovered that an extract from it was remarkably effective at killing those HeLa cancer cells in the lab.
That's incredible.
Yeah.
But how does it differentiate between cancer cells and healthy ones?
That's the truly remarkable part. This fungus extract seems to be more toxic to the HeLa cancer cells than it is to healthy cells.
So, like those other drugs we discussed, this fungus extract seems to have a preference for attacking cancer cells. That's a pretty big deal.
It is. It highlights the potential of harnessing the power of nature in the fight against cancer. This fungus, coexisting with the soursop plant, might hold the key to developing new, more targeted cancer therapies.
This is amazing stuff. We've gone from parasite medications to fungi on leaves, all in the quest for fighting cancer. It's like the future of medicine is hiding in plain sight. But how do we go from these initial discoveries to actual treatments?
That's the million-dollar question. It takes a lot of research to fully understand these compounds, how they work, their safety, and their effectiveness in humans. It's a journey that often begins in the lab.
So let's talk about that journey. What did the scientists actually do in these studies to figure out if these drugs had potential?
Take the nitazoxanide study, for example. They used kind of a two-pronged approach, combining in vitro studies, which use human colon cancer cells in a lab setting, with in vivo studies, which use a mouse model of colon cancer.
Okay, break that down for me. Why use both approaches? What's the advantage?
So in vitro studies are like a controlled environment where you can directly observe a drug's effect on cancer cells without the complexities of a living organism. But of course, we're not just treating cells in a dish, we're aiming to treat people. That's where in vivo studies come in. They provide a glimpse into how the drug behaves in a living system, how it's absorbed, distributed, broken down, and even how it leaves the body.
So it's about connecting what's happening at the cellular level to what might happen in a living being.
Precisely. In the case of nitazoxanide, they used a chemical called DMH to induce colon cancer in the mice. Then they divided the mice into groups, some receiving nitazoxanide and others not, to see how it impacted tumor growth.
And what did they find? Did it actually work in the mice?
The results were very promising. The researchers examined the colon tissues from the mice and found that those treated with nitazoxanide had decreased levels of certain proteins associated with that 1T-catenin signaling pathway we talked about earlier.
So it was like the nitazoxanide was putting the brakes on that pathway in a living organism, just like it did in the lab.
Exactly. But it gets even more interesting. Remember how I said nitazoxanide outperformed the standard chemo drug 5-FU in the lab?
I do. I wouldn't forget a detail like that.
Well, they compared the effectiveness of nitazoxanide to 5-FU in the mice as well. They determined the IC50 value, which, as you'll recall, is the concentration of a substance needed to inhibit 50% of a biological process. In this case, cancer cell growth.
Right. So a lower IC50 means a more potent effect.
Exactly. Nitazoxanide had an IC50 of 11.07 POM against the colon cancer cells, while 5-FU had an IC50 of 11.369.
So very, very close. But nitazoxanide still edged it out.
It did. And remember how we talked about the importance of a drug being more toxic to cancer cells than healthy cells?
Of course, less collateral damage is always a good thing.
They looked at that too. They found that nitazoxanide had a much higher IC50 value against normal colon cells compared to its IC50 against the cancer cells, meaning it took a much larger amount of the drug to impact the healthy cells.
Okay, that is promising. Less impact on healthy cells is a major win in my book. Now, what about ivermectin? How did they study its effects on those PAK1 proteins we talked about?
They used a combination of techniques, but one that stands out is called Western blotting.
Western blotting. Sounds more like something you'd order in a saloon than a lab.
It might sound a bit odd, but it's a powerful tool. It allows researchers to detect specific proteins within a complex mixture, like what you'd find in a cell.
So it's like sifting through a protein haystack to find the needle they're looking for.
That's a great way to put it. In this case, they were looking for PAK1 and other proteins involved in its signaling pathways within breast cancer cells. They found that treating the cells with ivermectin led to a decrease in the levels of active PAK1.
So the ivermectin was essentially interfering with PAK1's ability to do its job. disrupting those cancer-driving signals we talked about earlier.
Exactly. They also observed changes in downstream targets of PA1, further confirming that ivermectin was effectively disrupting these signaling cascades.
It's like a domino effect. Hitting that one crucial target, PAK1, sends off a cascade of events that ultimately disrupts the cancer cell's strategy.
A domino effect, orchestrated by nature and uncovered by science. It's pretty remarkable when you think about it. Now, it's important to note that while ivermectin shows promise in these preclinical studies, more research is needed, especially in humans, to fully understand its potential and any potential side effects.
Absolutely. We don't want to get ahead of ourselves here. But it's still exciting to see this kind of research happening. It seems like we're getting closer to a future where we might have even more tools in our arsenal to fight cancer.
I agree. And what's particularly encouraging is that many of these repurposed drugs already have established safety profiles for other uses. This can potentially speed up the process of getting them approved for cancer treatment if their efficacy is confirmed.
That's a huge advantage. Now let's talk about this fungus on the soursop leaves. How did the researchers even discover it had anti-cancer potential? They just stumble across it while strolling through a rainforest.
Not quite a stroll through a rainforest, but it does start with nature. They collected soursop leaves from various regions and then used a specific process to isolate the endophytic fungi, which are fungi that live inside plant tissues.
Endophytic fungi. Okay, that's a new one. What makes them special?
Endophytic fungi have a symbiotic relationship with their host plants. They live within the plant's tissues, and while they benefit from the plant, they also often produce compounds that help protect the plant from diseases or pests.
So they're like tiny microscopic bodyguards for the plant. And those protective compounds they produce could potentially benefit us as well.
Exactly. It's like nature's own pharmacy, waiting to be explored. In this case, they cultured these fungi in a lab setting to grow larger quantities and then tested extracts from them against several cancer cell lines.
And that's when they discovered its kryptonite-like effect on cervical cancer cells. What did those tests involve?
They used a method called the MTT assay, which is a common way to measure cell viability. Essentially, how many cells are alive and kicking after being exposed to a substance. The more potent the substance, the fewer surviving cells you'll have.
So it's a way to measure how effectively the fungus extract is killing those cancer cells.
Exactly. They tested it against HeLa cells, cervical cancer, and Y doctor cells, colon cancer, and compared the results to its effects on Chang's normal cells, which are commonly used in research as a control, to see how toxic a substance is to healthy cells.
And what did they find?
The results were quite interesting. The fungus extract showed significant activity against both the HeLa and Y doctor cancer cell lines, meaning it was effectively killing those cancer cells.
Okay, that's promising. But what about those normal cells? Did it nuke those too?
That's the crucial question, and that's where it gets really exciting. They found that the fungus extract was significantly less toxic to the Chang's normal cells compared to the cancer cells.
So like those other drugs we discussed, this fungus extract seems to have a preference for attacking cancer cells. That's a pretty big deal.
It is. It highlights the potential of harnessing the power of nature in the fight against cancer. This fungus, coexisting with the soursop plant, might hold the key to developing new, more targeted cancer therapies.
This is incredible. But we've got to remember this research is still in its early stages, right?
Absolutely. We need more research to confirm these findings, understand the long-term effects, and determine the best ways to potentially use these discoveries to help people. But it's definitely a promising step forward.
Okay, so we've got these exciting leads. But I'm curious, what are the next steps? How do we go from a promising extract in a lab to a treatment that could help people battling cancer?
The journey from lab to life-saving therapy is a multi-step process. It starts with identifying the active compound in the extract, the specific molecule that's doing the heavy lifting.
So like isolating the special ingredient in a complex recipe.
Exactly. Once they've identified it, they need to figure out its chemical structure and find ways to synthesize it in the lab.
Synthesize it. Why not just extract it directly from the fungus?
It's often more efficient and cost-effective to synthesize it in the lab, especially when dealing with large quantities. Plus, synthesizing it allows researchers to tweak its structure, potentially making it even more effective or reducing side effects.
So it's like taking a brilliant design from nature and fine-tuning it in the lab.
Precisely. Once they have a reliable way to produce the compound, the next step is preclinical testing. This involves testing the compound in cell cultures and animal models to assess its safety, how it's processed in the and of course, its effectiveness against cancer.
So it's all about making sure it's safe and effective before they even think about giving it to a human.
Exactly. If the preclinical studies are promising, the researchers can then apply to regulatory agencies to begin clinical trials in humans.
Okay, so let's talk about those clinical trials. They're like the ultimate test, right? Taking something from the lab and seeing if it actually works in people.
They are. Clinical trials are carefully designed research studies that involve human volunteers. They typically involve several phases, each designed to answer specific questions about the drug.
So walk me through those phases. What are the researchers looking for?
Phase 1 trials are usually the first time the drug is tested in humans, and they're primarily focused on safety. They're usually conducted with a small group of volunteers and aim to determine the safe dosage range and identify any potential side effects.
Safety first. What about effectiveness? What do they figure out if the drug is actually working?
That's the focus of phase 2 trials. These involve a larger group of patients who have the condition the drug is designed to treat. The goal here is to see if the drug has a beneficial effect and to gather more information about the optimal dosage.
So it's about seeing if it's doing what it's supposed to do in the real world, in people actually battling the disease.
Exactly. And then comes phase 3, the largest and most rigorous phase.
What happens in phase 3? What makes it so rigorous?
Phase 3 trials compare the new drug to existing treatments. They involve a large number of patients and are designed to confirm the drug's effectiveness, monitor side effects, and compare it to the current standard of care.
So it's like the final exam before graduation. If it passes phase 3, it's ready for prime time.
You got it. If a drug successfully navigates all 3 phases of clinical trials, demonstrating that it's safe, effective, and at least as good as, if not better than existing treatments, the company can then apply for regulatory approval to bring the drug to market.
Wow. That's a long and rigorous process. No wonder it takes so long to develop new drugs.
It is a lengthy process, but it's in place to ensure that new treatments are thoroughly vetted for safety and efficacy. It's all about balancing the potential benefits with any potential risks to patients.
I can see why that's so crucial, especially with something as serious as cancer treatment. Now, you mentioned that the researchers in the nitazoxanide study also took a close look at the colon tissues under a microscope. Why is that important? What were they looking for?
It's like being a detective, examining a crime scene. By looking at the tissues under a microscope, researchers can get a close-up view of the cellular landscape, searching for telltale signs of cancer and observing how the drug is impacting the cells.
So it's like zooming in on the battleground to see what's happening at the cellular level.
Precisely. They were specifically looking for histological changes, changes in the microscopic structure of the tissues that could indicate the presence of cancer and how severe it is.
And what kind of changes indicate cancer?
They were on the lookout for things like crypt distortion, dysplasia, goblet cell depletion, and hyperplasia, all signs that the cells are no longer growing and behaving normally.
Okay, those are some pretty complex terms. Can you break them down for me? What do they actually mean in terms of what's happening in the colon?
Imagine the lining of your colon as a landscape with valleys and peaks. The valleys are the crypts, and in a healthy colon, they have a regular, well-defined structure. But in cancer, these crypts can become distorted, their neat architecture disrupted. That's crypt distortion.
So it's like the cancer is messing with the colon's internal architecture.
Exactly. Now, dysplasia is like finding a rogue nation within that landscape. It refers to abnormal cell growth cells that are dividing too quickly, have irregular shapes, and don't look or act like their healthy counterparts.
So it's a sign that the cells are on the path to becoming cancerous.
It's considered a precancerous change, like a warning sign that things are headed in the wrong direction.
And what about goblet cell depletion? What are goblet cells and why are they important?
Goblet cells are like the lubrication crew of the colon. They produce mucus, which keeps the lining of the colon moist and protected. But in colon cancer, the number of these goblet cells often decreases.
So it's like the cancer is drying up the colon's natural defenses.
Exactly. And finally, we have hyperplasia, which is like uncontrolled urban sprawl. It refers to an increase in the number of cells in a tissue, making it thicker than it should be. In the case of the colon, this thickening can disrupt its normal function.
So by examining the colon tissues for these histological changes, the researchers could actually see the impact of the nitazoxanide.
Precisely. They found that the mice who received nitazoxanide had significantly fewer of these abnormal changes compared to the mice that didn't receive the drug. The nitazoxanide seemed to protect the colon from the damaging effects of the DMH.
That's amazing. So not only were they seeing an effect on those molecular pathways, they were seeing real, visible differences in the tissues themselves.
It's like having multiple pieces of evidence converging to support the same conclusion. Nitazoxanide might hold real potential as a weapon against colon cancer.
This is incredible. Now, earlier you mentioned that the researchers also looked at a marker called PCNA in those mice studies. What is PCNA and why is it important?
PCNA, or proliferating cell nuclear antigen, is like a construction crew foreman for cells. It's a protein that plays a critical role in cell division, ensuring that DNA is copied correctly when a cell divides.
So it's essential for making sure new cells are healthy copies of the original.
Exactly. But here's the thing. Cancer cells divide uncontrollably, like a construction project gone haywire. And because they're dividing so rapidly, they have much higher levels of PCNA than normal cells.
So it's like a red flag, a signal that cells are dividing too quickly.
Precisely. By measuring PCNA levels, researchers can get a sense of how aggressively a tumor is growing and spreading.
And how do they measure those PCNA levels in the mice?
They used a technique called immunohistochemistry. It's a way to visualize the presence and location of specific proteins within a tissue sample. In this case, they were looking for PCNA in the colon tissues.
And what did they find?
As you might expect, the mice that received DMH to induce the colon cancer but didn't receive any mitoxanide had a high number of cells with lots of PCNA, indicating that the tumors were growing rapidly. However, in the groups that received nitazoxanide, there was a significant decrease in PCNA levels, suggesting that the drug was effectively putting the brakes on the uncontrolled cell division.
That's really promising. It's like the nitazoxanide was telling those cancer cells to slow down and get back in line.
It's like it was restoring order to the construction site.
It's incredible how these researchers piece together clues from different levels, from molecules to cells to tissues, to understand how a potential drug might work against cancer. It's like putting together a giant jigsaw puzzle.
It's a fascinating process, and each study adds a new piece to the puzzle, bringing us closer to a more complete understanding of these diseases and how to treat them.
Speaking of piecing things together, I'm curious about the research on the soursop leaf fungus. They use something called ethyl acetate to extract those beneficial compounds.
Yeah.
What is that and why did they choose it?
So ethyl acetate is a solvent, kind of like a chemical that can dissolve other substances. Think of it like making tea. Hot water is your solvent, and it extracts the flavor and compounds from the tea leaves.
Okay, so ethyl acetate is like the hot water in this scenario, but instead of tea, we're brewing up a cancer-fighting fungus extract.
Exactly. But just like you wouldn't use cold water to brew tea, you wouldn't use just any solvent to extract these compounds. Ethyl acetate is good at dissolving a certain class of compounds, often those that are biologically active, meaning they can have an effect on living things.
So they're choosing a solvent that's specifically good at pulling out the good stuff, the
Yeah.
potential cancer-fighting compounds.
Exactly. And in the case of the soursop leaf fungus, it seems to have worked.
It definitely seems that way. This whole deep dive has been pretty eye-opening. We've explored the potential of repurposed drugs like nitazoxanide and ivermectin, uncovered the cancer-fighting abilities of a fungus, and delved into the science behind these discoveries.
It's been quite a journey through the exciting world of drug repurposing and natural product research. And we've only just scratched the surface.
What's really incredible is that these discoveries offer hope for new and potentially more effective ways to fight cancer. But if there's one thing I've learned today, it's that it's a long road from a promising finding to a treatment that can truly help people.
You're absolutely right. It takes rigorous research, careful testing, and a commitment to scientific rigor to ensure that new treatments are both safe and effective.
Absolutely. So as much as we love a good eureka moment, it's important to remember that science is a process, and it takes time to translate these discoveries into real-world solutions.
Well said. But it's a process driven by curiosity, innovation, and a relentless pursuit of better treatments and cures. And that's something worth celebrating.
Absolutely. This has been an incredible deep dive. Thanks for walking us through it. Until next time, keep exploring.
It's been my pleasure.